It's a real pleasure to introduce Knud Jonsson. Most of you know Knud. This is his second visit to the MVV. Knud is in his, remarkably only in his second year of PhD, and so this isn't a finishing talk, but he's published 5 papers to date, 2 in biology letters, 2 in MP, and 1 in Journal of Avian Biology. So he's a remarkably productive person. He's based, his home institution is University of Copenhagen in Denmark, and then he's co-supervised, so he's supervised there by Jon Fjeldse, and then co-supervised by myself here, and then Les Christenes at the Australian Museum in Australia. And I'll just quickly mention that Jon Fjeldse will be here from the 1st of June until the 12th of June, so if anybody would like to meet him, let me know. He's also willing to give a seminar if there's enough interest. So what Knut's going to talk about today is this really remarkable group of birds called the Crown Corvida, or crow-like birds for those less familiar. And I think what makes his work even more remarkable is many of these taxa come from really, really isolated ranges. So he's particularly concentrated on Malaysia, so many of these tiny islands. He's done field work in Australia, the Solomon Islands, New Guinea, and then last year went to try and get 2 of the world's rarest birds, the Mauritius and Reunion cuckoo shrikes, of which one species there are only 35 left, and another I think there are about 120. So he went to 2 tiny little islands in the Indian Ocean, and through various means we managed to get blood samples from both of those taxa. And I think what you really need to appreciate when he goes through his talk today is just how much of this data has also had to come from museum specimens, and the value that some of those specimens, some of them over 100 years old, are being able to provide for us to try and understand the biogeography of this very complex region, and then how much work goes into doing that as well. So,
all right, thanks very much. I wonder how much is left for me to say now. The headlines in there. Yeah, so I want to take this opportunity to thank everyone here, because it's been a real big pleasure to be here. Being here 3 months last year and 3 months this year, and time just flies. I mean, I managed to do some lab work, and I think could always use some more hours, but it's been a really great pleasure to be here. Everybody's so nice and helpful all the time. So yeah, so Rory's one of the supervisors, and here's another one. That's my main supervisor, Jon Fjeldse. I think some of you may have met him, and some maybe haven't. Jon has worked a lot in South America and in Africa, and I think he was quite happy with me starting to work in the Southeast Asian region, because that gave him a chance to sort of learn more about the birds and everything there. And I even had the chance to go in the field with him. This is in the Solomon Islands, and this is at the Welcome Committee, where Jon, as the boss, got the nicest headpiece of crown, and he looks a bit like God, I think. Jon is very innovative and has a lot of really good ideas, and a very good example of this is in the field. This is one of the most rainy areas in the Solomon Islands. It gets about 7 or 8 meters of rain a year up in the highlands in Makura, and we had a huge problem drying our samples. So Jon came up with this idea that we could dry them over this portable stove. And I don't know if you can see it, but there's a lot of like cotton and labels and a lot of stuff, and he was really happy. He was like a little kid, like, look at this, all the flies are going out and the skins are drying. And the next slide, obviously I was sitting with the back to him, and I heard this ooh, 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 and he threw all the samples on the ground, and our taxidermist was not very happy with all this. 2 days of work just burned within seconds. The samples didn't look too nice. But Jon, of course, is not down for a very long time. So we just invented a new mechanism, and this worked much better for drying our samples. So he's a pretty cool guy to be working with. So here's the official project title of my PhD. So it's determining biogeographic patterns of dispersal and diversification in Crown Corvida in Australia, Southeast Asia, and Africa. And I probably even could have added in the Western Pacific, because it's a pretty big region, and maybe the title is a bit long, but at least that's the whole area that it covers. And Crown Corvida, what is that? So here's Passeriformis, which is the largest avian order, and they're subdivided in two suborders, Subicines and Usines. And of these two groups, the Usines constitute the largest of the two groups. And then this is according to Siblin Alquist, and they split the Usines into three parvorters, Corvida, Passerida, and then this parvorter with some odd species which they didn't really know where they fit in. And so after they did their studies, I mean, several independent groups have found out that Passerida is monophyletic, whereas Corvida is not. So within these parvorters, they assign these superfamilies, and so these superfamilies are actually they don't form a monophyletic group. So I just want to show you a few photos of the birds. So this is one of the superfamilies Sylvioidae, swallows, songbirds, larks, huge group of babblers, and several other birds. Musicopoidae with nuthatches, dippers, waxwings, starlings, and Passerida with lots of finches with its. And then this weird group. So this group in Cercosphagus digitatidae includes two species of Digitatus found in forest areas of West Africa, two groups of sharetops endemic to South Africa, to dry highland areas, and then this Malaysian whale babbler which is endemic to Malaysia. So that's one type of fairly tiny group with no other birds in there, and it's pretty interesting to find South African, West African, and Southeast Asian endemics together as each other's close to close. So here's Corvida. Here's the basal most part of Corvida, Corvida. Lyrebirds of Australia, honey eaters of Australia and New Guinea, grass wrens the same, and fairy wrens, all Australian and New Guinea birds. And here are some of the Crown Corvidan birds. Birds of paradise, crow-like birds. This is another bird of paradise. And this is actually, it looks like it's upside down, and it is. Sort of, at least it's hanging upside down, because when these birds display, they fold out the wings, and they swing around these branches to impress the ladies. And they do that pretty well here. And now, here are my birds. So these are also within Crown Corvida. They include Shrikes and minivets, Chambophagidae. They include whistlers, Peggysophalidae, and Oriolidae, orioles, old-world orioles. So these are the groups that I'm working on. Cougar Shrikes and Cougar Shrikes are everywhere. Then Africa, then Asia, East Asia, South Asia, and Australia. And minivets are only in Asia, in Southeast Asia, and some of the islands, Borneo and the Lesser Sundas. And whistlers are Asian, Australian as well, and orioles are in Africa and Eurasia and Australia. So they're really good groups for finding out how did they disperse. We figure that most of these groups have their origin, or at least the Usines have their origin in Australia. So the big question is, how did they come out? How did they get out of Australia? And here's a study that was published in 2002, I think. So this group found out that Usines had their origin in Australia, or the passerine birds had their origin in Australia, and that Subicines probably went up through South America. There's a few old world Subicines, pitters and broadbills, and they might have come up through Africa. And then Usines went through Australia, and from there dispersed via Southeast Asia and to the rest of the world. So for my master's project, I'm going a little bit back in time now to give you an idea of the whole why I decided to work on these groups of birds. For my master's project, with Jon Fjelsø, we decided to do a super tree of all published phylogenies and Usine passerine birds. And we managed to construct a super tree with 1,724 taxa. And we covered pretty much all the families and subfamilies and tribes. So we had a good representation of all the birds in there. And just, yeah, I guess most people know what a super tree is, but it's just combining many smaller overlapping source trees. So here you can see that A and B are sister. There's no B in here, but A is sister to F, and the sister to these are C. So all you can do is you can combine them and say, all right, A and B and F are each other's closest relatives, and C is the sister to these. So you sort of combine them and try and get an idea of who's related to who.
Which method did you use to do that?
I used the manual method. I just went through 99 source trees and, you know, compared them and yeah. There are lots of different methods to do this. This is what we came up with. Yeah, so up here is Passerida, and down here is Corvida, and as you can see, these are what Sibley and Alquist considered a monophyletic group, all this, and as you can see, there's pretty good evidence that Corvida is not monophyletic. And if we highlight this group here, so here you can see Crown Corvida, and you can tell that there's pretty poor resolution among these groups. Here the cuckoo shrikes, whistlers, orioles, vireos are in here, which is pretty exciting. North American or American group of birds, along with all these Australasian bird groups. So this is the group that I pretty much decided to work on. This is a DBA analysis that we did for the data, for the super tree data. And the interesting part about this is the green is Crown Corvida. So we found out that dispersal of the origin of all these clades was Australian. So probably this group dispersed, or all these groups dispersed from Australia to Asia and the rest of the world. Whereas here is African origin for this and this node. So probably it could indicate that the whole Passerida group originated in Africa, from where it dispersed and undertook this massive radiation that it later became. So the biogeographic conclusions was from this study was that Passerida, Picotatus and Sharetops, crossed the Indian Ocean about 50 million years ago, and then underwent massive radiation, and that the Crown Corvida dispersed via Southeast Asia somewhat later, about 25 million years ago, and never experienced the same success, in the sense that there aren't as many species today as for Passerida. And when you look at a map, it makes some sense, because at that time Australia was much further south, and Africa was as far away from Australia as Asia was. So, I mean, if they might as well have gone to Africa as to Southeast Asia. And here's a map where you can see like land plateaus that probably at some point was above water. So here's the Brogan Ridge Plateau, here's the Kerguelen Plateau, and here's the Crozet Plateau. And they would have been in line when Australia was further south. So the birds, perhaps the birds could have island hopped across to Africa at that time, or they could even have used Antarctica gone through that way. Here's what the world looked like 14 million years ago. And now you can see that Australia is much closer to the whole Southeast Asian region. And you can imagine that birds would have had an easy time jumping across islands to some across to Asia. So overall, the reason for studying Crown Corvida was it's a poorly studied group, probably reflecting that it's been difficult to access fresh material. Cuckoo shrikes are canopy birds, and they're really, really hard to catch in nets. I've tried on 2 occasions. In the Solomon Islands, when doing field work, we got one cuckoo shrike sample, which was slingshot by a local boy. We didn't get any in the net. And in Mauritius, I tried also to catch cuckoo shrikes, and they'll move around in the canopy, come down a little bit every now and then, and they'll pick out little spiders or insects. So when they come to the net, they simply see the net and they just go over the net and keep going. I've even seen birds like flying really fast through the forest and just come to the net and go over it, and keep flying. So, tricky business. So a lot of the work that I've done has been from towpats, and only, regrettably, too few samples from fresh material. And then, of course, the very exciting region, complicated tectonic movements, and it's all about working out how could these birds have moved across. Were some of them ferried on these fragments of land that moved from Australia to Asia? So here's a map where you can tell that here's Sulawesi. This is actually an old map, but you can tell that half of it is of Australian origin. All the this sort of like dotted color means that it comes from Australia, and the rest is from Asia. This is not really the true because there's a Philippine plate coming down here as well. So Sulawesi consists of land masses of 3 different plates. So the Philippine plate, the Asian plate, and the Indo-Australian plate. And I just brought this cartoon so you can see what happened. Notice here how Australia came from the Gondwana supercontinent and started moving northwards here. These pieces become Sulawesi at some point. And you can really, there's an island arc that moves out and comes back in again. And it's really interesting to see how the whole thing, when the land masses collide, they start turning in a counterclockwise fashion, and all the little islands of current Indonesia becomes like mixed together. There's another part of Sulawesi moving southwards, and up there's the time. It's 1.3 million years ago. And now it starts moving around. So it makes sense around this time to think that birds that live here could have started island hopping across. And if some of them were even quite sedentary, they could have been ferried on these pieces and come to Sulawesi. And obviously Sulawesi is a major island that has components of both Asian and Australian origin. Now you can see not so long ago it's pieced together. It becomes what it looks like today.
You can see the arcs including the northern New Guinea.
Yeah, that's yeah. So that that's pretty interesting, and I've been in contact with some of the big geologists in the region, and every time I write them, they're very nice people, and they go, it's really hard to give you. We'd like to have dates for various islands so we can date our phylogenies, and they're like, it's really hard to tell what was above or below water, or what was even at a certain place. So, but I think I'm getting closer. This is the first sort of major paper on all the groups that was published. You can see there's a lot of people in there, and that was just getting the overview of the relationships of the different families. And the most interesting part that we found out in this paper was that we found 3 independent sister relationships of Asian and African birds. So these 2 cuckoo shrikes, the pygmy cuckoo shrike from Sulawesi and the I forget the common name on this one. But this is an African cuckoo shrike, and they're each other's closest relatives. And we had for this study we had a fairly good sampling. We still lacked some samples, and I've done those in the lab this time. So, of course, we're curious about, does this really hold up when we have all the other cuckoo shrikes in here? But we have several other species from the mainland, and Borneo and Australia and New Guinea. Here's another example of a sister relationship between New Guinea and Africa. And here's the third example, again including Sulawesi, which is interesting. 2 examples including Sulawesi, and Madagascar, a species from Madagascar. So of course the next step here, the first question you always get when you postulate long distance dispersal is, what about extinctions? It's the first thing everybody puts in. We always get that comment, couldn't they just have been everywhere here, and then they died out in between? And if this was 25 or 30 million years ago, a lot of extinctions could have taken place. Of course, nobody knows, but at least one crucial sample we lacked was the Indian cougar shrike, so we have that one now. And so that's the next step to find out. So another step was to find out whether these island groups. The problem is there's nothing in between, so you can't really work out if they just jumped across because there's nothing left today. But at least we were interested in finding out here's Mauritius and here's Reunion, and they both have an endemic cougar shrike. So of course we're interested in finding out where they belong. It would be obvious to expect them to just be of Madagascar, African origin. So that was a pretty cool project, I thought, that's great, I got a chance to go to Mauritius. And so I went, and I was in the forest with my mist nets trying to catch cougar shrikes, and I caught the endemic pink pigeon, which is, they have a whole rescue program for that one. All the volunteers were not very happy with me. And I caught one of the endemic parakeets. They've all been down to numbers of like 5 or 10, and they have this whole big rescue program. And I'd put up my nets around in the forest, and I'll catch everything but my cougar shrikes. So the volunteers would come up to me and say, you know, is there any other way we can do this rather than you disturbing our rare birds? And so I found a nest, and there was another group there looking for cougar shrike from the Reunion program, and they found a couple of nests as well. And so we decided that when the eggs would hatch, they would climb the trees and they'd take a blood sample from the chicks. So that's what they did. And actually, it's very sad, all the cougar shrike nests in Mauritius that year were predated. They don't know by what. So, I mean, hey?
Not by you.
Not by me, no, not by me. And the first nest that I found, the first 2 nests that I found were predated before they even got youngs. So they didn't even get a chance to climb the trees before they were predated. And this nest was predated, I think, a couple of days later, and all the other nests as well. So there was a 0 success rate for the Mauritius cougar shrikes. They told me in Reunion they had a high success rate this year. So, frankly, if you manage the population, you can do something. This is actually, this forest area is fenced off, so they try to keep out all pigs and ferrets, but it's pretty much impossible. They have traps everywhere. In any case, here are the Madagascan cougar shrikes. And that's the Mauritius and the Reunion cougar shrike. And the studies that I've managed to sequence 4 genes for these cougar shrikes now, 3 nuclear and 1 mitochondrial. And with a quick and dirty neighbor joining analysis, for all 4 markers they go with Asian trillers, the larger trillers from this region over here. So that's pretty interesting. They don't go with the African birds, so that's a 4th example of a sister relationship between African-ish and Asian species. I thought that was pretty exciting. So I'm looking forward to starting analyzing more and digging more into this. All right, so enough about the cougar shrikes. This is a study we did on the golden whistler complex of Pachycephalo pectoralis. It's a pretty common bird in Australia, and in New Guinea it's pretty much absent. But then it's out on all these islands. This is the Solomon Island group. This is the Bismarck Islands, the islands east of New Guinea, New Ireland, New Hanover, New Britain. And in this study, this is Jon's painting here. There's 3 species pretty much that look the same with a white throat. And it's pretty interesting to see that this one up here, Pachycephalo melanura, is found on tiny little islands. I don't know if you can read them, but I'll show you a map. So the top one with the white throat is found on these little offshore islands. You can't even see them on the map. Here's an arrow, Kretna, Tingwin, and Kum, small offshore islands. And the closest relatives are found in eastern Australia, southeastern Australia, and it's this one here. There's even this one up here, there's even one in the Northern Territory in mainland Australia, in the mangrove area. So we have a group of birds on small offshore islands off the eastern part of New Guinea and in the Northern Territory in Australia. and they're closely related to eastern Australian birds. And then in turn, those two are related to Western Australian golden whistlers. So golden whistle, and this has, I didn't put the support values, but this is well supported up here. There's another one that looks pretty similar, and that's this one. And that taxon is found on the large islands here, New Britain, New Ireland, New Hanover. So it's pretty interesting to see how that, an example of, probably, we're guessing that this could be an example of a supertramp species. And Jared Diamond, for Jared Diamond, this was one of the classic examples of a supertramp species in Melanesia. So it seems like maybe they just move around between little islands. They've been seen on the main islands, but they're not known to breed there around all the time, and they can only maintain populations on these small islands. Perhaps somebody has looks like a face up something like. Here's that's another study that I'm working on. I haven't come very far with it yet, but this is a study on all the pachycephalo whistlers that we have, which includes Pulvicinclus shrike thrushes, if you know those. And this is we've tried to plot in how many species are on each square here. We didn't at that time. Australia wasn't done yet, so there's quite a few species down along the east coast of Australia, and even in the north and west as well. But probably the highest diversity would be around here, and then in New Guinea. And I'm not really sure what to do with this data yet, but I think it's interesting that the highest amount of species are up in the highlands in New Guinea, and then it looks as if there's a pretty rapid radiation taking place. And I'm curious, I hope I get the chance to talk to you about this, Craig, at some point. What could cause this, and I'm looking forward to use some more sophisticated analysis methods on this. But this is as far as I've come on this, so not very far. And finally, I just want to show you this one, which is a phylogeny. When we decided to do whistlers, usually pirahuis are thought to be in there. So when I started sequencing last year, I was sitting with the data, and I said to Rory, that's really funny. Why did the pirahuis don't come out together? I didn't even think about it. I was just doing like a neighbor joining analysis, and Rory says, really? That's interesting. And when we did the proper analysis, we had 5 species of the 6 pirahui species, and you can see that they come out in various places in the tree. So one comes with the actual whistlers. One goes with the shrike thrushes, which assisted to whistlers. 2 down here go together with orioles, one-wheeled orioles, and one is just far away. So I just want to show you what they look like. So these are 4 of the pirahui species. These 2 look kind of similar, but this could be anything. This is just a brown bird. This is another brown, but I mean, this is another brown bird, right? So it's, I don't know why they've been put together in the first place. So these 2 actually go together and are sister, and I'm not sure what could be in between, but it seems that there's nothing really in between, and that they're sister through this orioles. So obviously that's what I've been doing here this time as well. I've been sequencing pretty much all the oriole species of the world. So it'll be interesting to see I'm pretty sure they're outside orioles, but the question is how close to orioles are they? So here's the crested pirahui, which is close to this, another art species which was previously thought to be a whistler, and which we found out is not. So they go together in one group, and it's not really close to anything. Well, close actually to like a whole group of cuckoo shrikes and African bush shrikes. And finally, here's that odd brown bird, which is close to shrike thrushes. And I mean, these are all like sand-colored, mostly sand-colored birds. But I mean, I think if you look at them, maybe you could say, maybe they do look a little bit like them. I don't know, it's tricky. The interesting thing is also that about 10, 15 years ago, pirahuis were found to be the first group of birds that are toxic. And it's interesting to see now that they come out in 4 different places So toxicity is not limited to one group of birds. It's actually in several places within the corvid birds, which I thought was a pretty interesting finding. So I couldn't find a picture of this one, but that's the black pitahui. That's the only one that's actually a whistler. That's what's previously thought. So, and I think if you look at it and take away the breast spot and the bright colors, and yeah, you have a whistler. Yeah, so that's pretty much all I have to say. I want to thank you all very much again. It's been such a pleasure being here. So, questions?
Do you think then that there are a lot more toxic species of birds, non-pituites, that are toxic?
Well,
Or
I
do you
think
that it just
I
arose
think
many times?
we think that it's related to the diet. So it's probably a matter of I mean, they get it from caterpillars probably, or beetles. So yeah, a lot of caterpillars are toxic as well. So perhaps they could pick it up from certain prey objects. So it's probably just a matter of if orioles picked up toxic beetles or caterpillars and they could excrete it, I think a lot of birds would have could excrete toxic stuff on their feathers. I think it's possible. It'd be interesting to talk to people who catch a lot of birds in New Guinea at least. See if they ever experience like sneezing or numbness in their fingers or.
It's not detectable in dried skins.
Yeah, I think Dumbacher, who did the study, I think he used skins as well. But they found out in the first place by handling the birds in the field, and they sensed this numbness in the fingers, and their eyes were running and sneezing and that sort of thing. So perhaps, but a lot of people have done field work in New Guinea, so I don't know. Yeah, I don't know. I think it's possible. Yeah.
Well, why be toxic if you're not going to advertise?
Well, that's another. Well, we're moving into a whole other field. Yeah, I think that Dumbacher actually thought that they advertised it, and that there was some mimicry even within some of the pitahui birds.
Brown ones?
Yeah. But as far as I remember, there are different levels to how toxic they are. So I think these two were actually. Yeah.
I think they're the very toxic ones.
Yeah, I think these are the most toxic birds, and they actually somehow do advertise it, I would say. But yeah, being brown is not really. Yeah, I don't know. I haven't thought too much about that.
Could they advertise it in a color we can't perceive?
Sorry?
Could they advertise it in a color we can't perceive, like in another spectrum? So who are they toxic to? Well, Madagascan species, other animals we use in control. Controls and stuff. Or other things.
Yeah, I really don't know. Yeah, great.
So look, to come back to the main topic before. So when you see these sort of jumps across to Africa,
Yeah.
do you see a sort of burst of, do you see a sort of radiation?
Well, see, that's the next step we're going to do is a whole Kugelschreiber phylogeny. So get all the texts in there and try and find some calibration points and try and see when did this happen. But I mean, from the very preliminary data, it looks as if it could have been around the same time. Is that what you mean?
Yeah.
So, yeah.
And then once they get there, do they radiate quickly, or does this look like
The
normal?
thing is, they you know, they don't really radiate. That's the thing. It's kind of like it's just kind of like, you know, one species that. So this one. But this is. So here's the Cumberfaga complex, which is 4 species. This is one species up there. Cambochera is like the only one of its genus. So it's
I guess what I'm getting at, what's the structure of the phylogeny look like? Does it look like very closely branched divergence events
these.
among the ones that have jumped across? Or
I only have 2 species of this complex, and it doesn't look very tight knit. But again, I had 3 nuclear markers, and I need to add mitochondrial as well to see what's happening. But I think I need more species in here to be absolutely sure what's going on. But I mean, that would be really interesting if it happened at the same time. Yeah. But it's not like they radiate madly afterwards, after arrival in Africa. Yeah.
I guess you used a lot of topads in your studies.
Yes.
Can you tell us anything about, or did you learn anything about how the DNA deteriorates over time in the topads?
Do you mean if there would be any changes?
Yeah, and were the older ones harder to get stuff out of, or not?
Oh yeah. The older it gets, the harder it is.
So
that's the general rule. But
how old were you able to get stuff?
I can get stuff out. I've got stuff out of 1890, something like that. But it varies a lot. I mean, they have in Stockholm. They have stuff that was collected in the 30s, 1930s, and it all works really, really well. And then sometimes you get stuff from the 60s, and you can't get it to work. So there must be something how the samples were kept or stored, or maybe how fast they dried in the field, or I don't know. But there's definitely differences between collections.
Maybe if Jan's roasting method.
Yeah. yeah. I wonder. Yeah.
Okay, any other questions? Well, if you join me in thanking Knud, and good luck with the next step.